Hello and welcome to the first video for the Higher Human Biology course on the first topic of Unit 1, which is division and differentiation in human cells. So the first thing we need to establish in this topic is the two main types of cells that we have in the human body and a little bit about how they divide and their various features and characteristics. So we'll start off first of all with the somatic cell. So the somatic cell is any cell in the body other than the cells involved in reproduction. So that'll be your liver cells, your brain cells, your muscle cells, all of the cells that make up your body other than those involved in reproduction. And we'll talk about those cells in reproduction in a moment or two. Okay, so try and get familiar with that word somatic cell. A somatic cell divides by the process mitosis, which you should all be familiar with from National 5, to form more somatic cells. During cell division, the nucleus of the somatic cell divides by mitosis to, and this is a phrase you'll be familiar with from that 5, maintain the diploid chromosome number. Okay, and that is a phrase it's going to be well worth you getting familiar with. Okay, maintains the diploid chromosome number, which essentially means it keeps the same number of chromosomes in the daughter cells that we find in the original cells. Okay, in humans, because we're obviously focusing primarily on humans in this course, it's going to be 46 chromosomes, but those 46 chromosomes are arranged in 23 pairs. Okay, and we can see from this diagram at the bottom here, that arrangement, okay, we get 23 pairs, and we call them homologous chromosomes because if you look at each pairing, you'll see that each chromosome looks very similar to the other one in the pairing. Okay, but then if you look at the pairs compared to other pairs, you'll see they are different from each other. And that's because you inherit one of each pair from your mother and one of each pair from your father, okay? So we say diploid cells have 23 pairs of homologous chromosomes. That's 46 individual chromosomes altogether. And you'll notice that last set of chromosomes there, which are either XX or XY, depending on whether you are female or male by sex, okay? So and of course, there are other variants than XX and XY, but that's beyond the scope of the course. So we're not going to go into too much detail there, but you can always do some extra reading and research on that if you wish. So the second set of cells that we need to know about are the germline cells. So the germline cells are the gametes, that's the sperm and ova, and the stem cells that divide to form the gametes. Now, a common mistake that people make here is when they're asked about germline cells, they usually remember the gametes, but they forget that the cells that divide to form the gametes are also germline cells, okay? So any cell involved in reproduction, not just the reproductive cells. Now, germline cells divide by mitosis to produce more germline cells, okay? Just in the way that we've learned about previously, or by a new process this year called meiosis, and that's to produce haploid gametes, okay? Now, at higher, we don't have to go into the process of meiosis in too much detail, but we do need to know about a couple of the steps. So first of all, the nucleus of germline cells can divide by meiosis instead of mitosis. Okay, so when a germline cell is looking to create a sex cell, a sperm or ova, then it will divide by meiosis. Okay, it undergoes two separate divisions. The first set of divisions involve separating the homologous chromosomes. Okay, so they line up at the equator as homologous chromosomes, and those two in the pair get separated to opposite poles. Now, the second stage is one that we're a bit more familiar with, where we are going to be separating chromatids in a similar way to how we did that in National 5 and mitosis. Okay, so there's two divisions, firstly separating homologous chromosomes, and secondly, separating chromatids. And lastly, haploid gametes contain 23 single chromosomes. Now, our diploid cells had 23 pairs of chromosomes, or 23 homologous chromosomes, but the haploid gametes contain 23 single chromosomes. Okay, so just one from each pair compared to the diploid cell. So that's the difference between the two types of cells, the somatic cells and the germline cells that you need to know about. Now, shout out here to a brilliant YouTube channel called the Amoeba Sisters. If you haven't yet subscribed to them on YouTube, I would suggest going and finding them just now and doing that. They produce brilliantly animated videos on a wide variety of biology topics. Now, The one word of warning I would give is that they are set up to primarily serve an American market rather than a UK or and specifically a Scottish market. So if you're sitting a Scottish exam, it will mean that you have to be selective about the content in terms of what you learn from it. I think that what they're excellent for is like a good overview of topics and processes and to visualize it and see it in animated form. But sometimes, the details they go into are different from the details that we have to go into as part of the Scottish qualifications. So keep that in mind, but do go and watch some of the videos there. They are great to watch and they're a brilliant channel. So moving on, cellular differentiation. Now, cellular differentiation is the process by which a cell expresses certain genes to produce proteins characteristic for that type of cell. Okay, now I would suggest learning that entire definition. It's a very common question in the past paper questions where it asks you to define cellular differentiation. Okay, and so knowing that definition is going to get you marks in all likelihood in your exam. Okay, so it's the process by which a cell expresses certain genes to produce the proteins that are characteristic for that cell type. So, for example, a red blood cell, it will not express the gene for a nucleus because does not have a nucleus. However, it will express the genes that will give it a biconcave shape. Okay, so it only expresses the genes that will produce proteins that are characteristic for that cell type. And that's going to vary depending on the specific cell type that the body is looking to differentiate into. Okay, and this allows the cell to carry out whatever specialized function it has. And there's a wide array on the diagram there on the screen that you can see. Now, all cells tend to be different in terms of their appearance, and usually when structure is different in cells, the function is also different. Okay, so the way in which they are built, the way in which they are differentiated will have a determining factor on their function. Okay, stem cells are the wonders of biology. They are unspecialized somatic cells. Now, they can do two things. They can either divide and make copies of themselves, which is handy, or differentiate into specialized cells. Okay, so make more copies of themselves or become specialized cells. Okay, and both of those functions are important for stem cells because throughout the body, there are a few areas where we have stem cells and we want to maintain that stem cell number. So when some differentiate, others will have to divide to maintain that stock of stem cells that we have within the body. Now, the two types of stem cells that you guys need to know about for your exams are tissue or adult stem cells. Now, they're involved in the growth, repair, and renewal of the cells found in that particular tissue. Okay, so we call them multipotent. Okay, remember that word multipotent. And what that means is that they can make all of the cell types found in that particular tissue only. So, for example, cardiac stem cells, ones found in the heart, can only make the cells that are found in the heart. Similarly, blood stem cells can only make the cells found in the blood. Okay, and it gives you the example at the bottom there. Blood stem cells located in the bone marrow can give rise to red blood cells, platelets, phagocytes, and lymphocytes. And we know from National 5 those are two types of white blood cells. Okay, so the blood stem cells can only produce cells found in that particular tissue type. And that's an important distinction to remember because in a moment we're going to look at a different type of stem cell which doesn't have those limitations. So the cells in the very early embryo or embryonic stem cells can differentiate into all the cell types that make up the individual and therefore are pluripotent. Okay, so you could take stem cells from this embryo and they would potentially differentiate into any cell type from that individual. Okay, so they have a much wider range of cells that they can differentiate into compared to the tissue ones. That's because all the genes in embryonic stem cells can be switched on. Now, they won't all be switched on. Okay, that would be chaos if you switched on all of the genes, but they can switch on any of the genes. Not all of the genes, but any of the genes. Okay, so these cells can differentiate into any type of cell. Okay, they can switch on the genes that make it into a skin cell. They can switch on the genes that will make it into a liver cell, or they can switch on the genes that will make it into a neuron. Okay, depending on the cell type that's required, embryonic stem cells can produce any of those cell types. Now, again, shout out to the Amoeba Sisters. They have a good video on exactly this process, okay? I'm not going to play the video within a video, but I'm sharing the image so that you can go and find this video on YouTube and watch it. It'll be well worth it. The animation alone is excellent, and they also describe things in good detail and give lots of nice examples in context. So do go and check out the Amoeba Sisters after you've watched this video. Okay, so the therapeutic uses of stem cells. What can we use them for in therapy and treatment and in helping people? Well, we use them in the repair of damaged or diseased organs or tissues. Okay, and we're going to look at a couple of examples of those. For example, the corneal repair that we can see in the first diagram there. Now, the cornea is the outer layer of the eye there, and when there are issues with that, they can actually remove that part, put in a graft which has been grown from from stem cells, and replace the cornea in that way. They can also regenerate damaged skin. So, for example, if you have burnt your skin with fire or acid or something similar, normally that would form scar tissue. But if you put a skin graft on it, which has been grown from stem cells, then that will help to repair that damaged tissue. And there's a lot of exciting things potentially coming up in terms of the therapeutic uses for stem cells. I'm a type 1 diabetic myself, and I know that they're doing a lot of research into potentially using stem cells to regrow the islet cells in the pancreas that secrete insulin. That's very exciting for me personally, but there are so many different studies going on for a whole wide range of diseases and conditions that will involve the use of stem cells to potentially repair those issues. So that's a very exciting time to be in, and hopefully as young people, as you become adults, you will reap the benefits of all of that research. And on that topic, it's worth noting that stem cells from the embryo can self-renew, which basically means that they're able to make copies of themselves if we put them in the right conditions in the lab. And researchers have obviously got that down to a fine art, because when you've got a supply of stem cells, you want to maintain that supply of stem cells so you can further your study and research, and you can continue to look at ways in which they can be used for therapeutic uses, for example. Okay, and I talked about research a moment ago involving the use of stem cells being used as, for example, model cells to study how diseases develop, or been used for drug testing. And of course, the reason they do that is because ethically, it's I guess more acceptable to use cells in the lab and program them to be diseased and then test drugs on them than it would be to test drugs on human subjects who have the disease. And for a number of reasons, because a cell just has there are less variables, there are less factors involved, less risks, if you like. Whereas if you are testing drugs on a full human, then humans are very complex and multifactorial. And so while the drug might be effective on the specific area that you're targeting, it might have other side effects. So it's good to test these things on cells first so that we can study them a little bit more, understand them better before we then start using these drugs on humans, okay? And for example, if you want to study a degenerative disease, for example, you can program those model cells to replicate what happens in those diseases, and we can see exactly what's happening inside the cell in a lab in the way that you couldn't if that disease cell was inside a human, of course. Now, stem cell research provides not just the model cells, but also provides information on how cell processes, such as cell growth, okay, and learning about how cells grow is so important, especially for conditions like cancer, for example. Differentiation, which is essential for us to understand how specific cell types are made from stem cells and how they're able to specialize. And gene regulation, so how genes regulate or express and then become the various proteins that make up an organism. Okay, how genes work in coordination with other ones, when are they expressed, etc. Okay, so there's lots and lots of cell processes that we can learn more about as a result of stem cell research. As I say, it's a very exciting time to be involved in stem cell research. There's so much new stuff being discovered all the time. However, all that said, there are many ethical issues with using stem cells. The use of embryonic stem cells, as I've already mentioned, can offer effective treatments for disease and injury, and there are loads more in the pipeline for the likes of Alzheimer's, dementias, type 1 diabetes that I mentioned before. However, it should be noted that it does involve the destruction of embryos. Okay, and that's obviously the drawback. Okay, that's the downside of using the embryonic stem cells. So effective treatments, but it does destroy the embryo. Now, the last wee section on this topic deals with cancer cells. So we're going to look at the biology of cancer and how it forms here, okay, as opposed to talking about all the other factors surrounding this condition. So cancer cells divide excessively. Okay, they divide excessively. That means they divide at a faster rate than they normally would. Okay, our cells divide and they divide fine. And in a way, cancer cells are not too different from normal cells. The main thing is they are not responding to our normal regulatory signals. Okay, those are signals which will tell the cell, hold on, you're dividing a little bit quickly, so let's slow down a bit. Okay, but the cancer cells ignore or do not respond to those regulatory signals and therefore begin to divide excessively. Now, the result of that is a mass of abnormal cells, which we call a tumour. And you're probably familiar with that word tumour. Okay, it's a very scary word. But that is what it means. It means it's a mass of these abnormal cells that have divided excessively because they haven't responded to the original regulatory signals that a normal cell would respond to. Okay. Cells within the tumour can sometimes fail to attach to each other and unfortunately can then spread throughout the body where they may form secondary tumours. Okay, and you've probably heard, in general chat, maybe family members with people on TV or programs or films, you've heard them talking about cancer spreading. And that's what this means. The cells within the tumour fail to attach to each other. And when they spread throughout the body and find a new place to locate, they can form these secondary tumours. Okay, and that's cancer spreading throughout the body. Okay, folks. So I hope that lesson's been useful for you. And that we've covered everything that you need to in that first topic. And I'll see you all for the next lesson. Hi there, folks. Welcome to the past paper questions for the first topic in higher human biology. So we're going to be looking at some past paper questions on division and differentiation in human cells, just to give you a little bit of practice to apply that knowledge that we've just gone over in the video. So question 1 here. So I have these booklets where I go through each topic by topic and I get all of the past paper questions I can find and I put them together, bundle them together, because I think it's good to have that sort of intense workout of that knowledge that you've just covered, just to apply it in the way that it would be in an exam. So number 1 here, it says, and just to say before we start, actually, if you want to answer the questions as we go, then pause the video after I've read the question and you can have a go yourself and see if you're right. So in a developing embryo, tissues such as muscle and nerve are produced by, so tissues such as muscle and nerve, are they going to be somatic or germline? Well, we know that all the cells in the body, apart from those involved in reproduction, are somatic. So we're going to go with somatic. And we know that somatic cells only divide by mitosis. Okay, so that narrows down our answer to C there. Okay, can't be A because somatic cells don't do meiosis and it's not germline cells. So we're looking for C for that first one. Next one, which line in the table best describes cell division in a specific cell type? Okay, so if we look at options A and B there, we've got somatic cells again. Now, the type of cell division it describes is meiosis. And again, somatic cells don't undergo meiosis, so we can immediately eliminate options A and B there. If we look at germline cells, we know that germline cells can undergo both mitosis and meiosis, so it could be C or D on that basis. But then we look at this chromosome number in the cells produced. Now, if a germline cell is undergoing mitosis, mitosis maintains the diploid chromosome number, so therefore the answer is going to be D for that one. Okay, if it said germline cell undergoes meiosis, then haploid would be right, but in this case it's mitosis, so we go with D. Germline, mitosis, diploid. Next question, you can see from the grainy fuzz in that question, it's an old one, but still a useful one to have a look at. Which of the following is not a use of stem cells? Okay, so we covered that skin grafts are a use, they're a therapeutic use. We talked about drug testing, which is in the use of model cells, okay, during research. We talked about bone marrow in terms of the tissue stem cells, but we did not mention IVF treatment. Further down the line in Unit 2, we'll talk about IVF treatment, but for now, we have not learned it, and it's not one of the uses that we're interested in just now. So it's going to be C, IVF treatment is not the use. Next question, stem cells in the red bone marrow. So the bone marrow gives rise to what kind of cells? Well, it gives rise to both red and white blood cells, the lymphocytes and the platelets, the phagocytes, as well as the platelets. Okay, so the answer is going to be D. Now, although C has two correct answers in it, it says only and excludes the white blood cells, so we need to include them. We know that B is only red blood cells. So again, we know that there's more than that the bone marrow will differentiate into. So it can't be that. And similarly with A, we know there's more than just the platelets that will be differentiated into. Okay, next question. Stem cells in the red bone marrow, are they A, already differentiated? They can differentiate into all cell types. They can only differentiate into bone cells or can differentiate into platelets and lymphocytes. So we know that it can only differentiate into cells of that particular tissue type. And we know that red bone marrow gives rise to the blood cells, don't we? So it's not going to be A or B or C. So the answer has to be D there, into platelets and lymphocytes. Okay, the next question 1 here, it says, which of the following descriptions is correct? Germline cells can only undergo meiotic division. Well, we know that germline cells can undergo both mitosis and meiosis, so that's wrong. Somatic cells undergo mitosis to produce haploid cells. Again, we know that mitosis produces diploid cells, so that can't be right. Somatic cells undergo meiosis, so we can stop right there. It does not undergo meiosis, so we can miss that one out. And then D, stem cells are somatic cells, which are relatively unspecialized. Now, we know that stem cells are unspecialized, even from our Nat5 knowledge. So the answer there is going to be D. So there's some multiple choice questions just to get us warmed up. So let's have a look at a couple of section 2 questions, which tend to be obviously the trickier ones for us. So this question says, the human body contains many specialized cells, all of which have developed from stem cells in the early embryo. So we've got nerve cells, we've got liver cells, we've got cardiac muscle cells. We need to name the process by which a stem cell develops into a specialized body cell and explain how this process occurs. So the process by which it develops into specialized cell is called differentiation, okay? Or as we call it, cellular differentiation, okay? So the process, cellular differentiation or differentiation would be acceptable. And how does this occur? Explain how this process occurs. So we would talk about that specific genes are being expressed to produce proteins that are characteristic for that cell type. And when I said earlier on about memorizing that definition, this is exactly the kind of question that would be useful for, okay? So it expresses the genes, the certain genes that produce proteins that are characteristic for that specific cell type, okay? Part B, both germline and somatic cells retain the ability to divide. State the type of cell division that only occurs in germline cells. So we know that somatic cells divide by mitosis. We know that germline cells also divide by mitosis to produce more germline cells. But we also know that germline cells undergo meiosis to produce haploid gametes. So the type of cell division that only occurs in germline cells is meiosis. Part 2, explain why mutations in germline cells are potentially more serious than mutations in somatic cells. Well, if we have a mutation in a germline cell, that mutation is going to end up in our sperm or egg, which is potentially going to end up in our offspring. So they can be passed on, the mutations, to offspring. Whereas in somatic cells, they will not end up being gametes, which will form offspring. So therefore, it will only affect the human in which the mutation first occurred. Okay, so the answer here is that mutations in germline cells can be passed on to offspring, which is why they're potentially more serious than the mutations in the somatic cells. Part C, companies developed a drug that could be used to treat the symptoms of an inherited disease. Before proceeding to clinical trials using volunteers, the company decides to carry out additional tests in the laboratory using stem cells. Okay, so describe one ethical consideration that might have influenced this decision to use stem cells. When we test drugs on humans, we know that it's humans are very complex and multifactorial, so it can be a risk when we test drugs on humans. And for most pharmaceutical companies, they have to go through so many hoops in order to be able to get to the stage where they do drug testing in humans. By using stem cells, they're going to be able to eliminate that risk to humans. Okay, so there will be no humans harmed in this procedure. So probably what they have considered, what would have influenced that decision, is the fact that there's no need to use humans to test the drugs. They can do it on the stem cells. There's less risk to humans. Okay, or something along those lines, a consideration like that. All right. So that's something that they probably would have considered. They might also have considered, to be honest, about the use of stem cells, i.e., the fact that it results in the destruction of the embryo as well. Is it right to destroy the embryo in order to use these stem cells? So, therefore, that would have been another perhaps ethical consideration that would have influenced their decision there. Okay, so that was a question there, and hopefully that section 2 question seems straightforward after going over it. This one, the diagram represents an embryo in the early stages of development and identifies the inner cell mass, which is made up of the stem cells. Okay. So state one feature of stem cells. Stem cells are relatively unspecialized cells, and they can go on to differentiate into many different cell types. So either of those features would have been acceptable. Okay, the fact that they're unspecialized, or they have the ability to differentiate or self-renew or divide to make more stem cells. All of these things would be features of stem cells. Part B, stem cells are also found in tissues throughout the adult body. Explain how the function of tissue stem cells differs from that of the stem cells found in the inner cell mass of an embryo. So for this question, there's a particular way to answer it, and students often don't quite get this mark, not because they don't have the knowledge, but because of the way they set out their answer. So here's how to do it. You would say that tissue stem cells are they're multipotent, or they have the ability to only differentiate into cell types of that tissue. Whereas, and this is the bit that students always forget, you've got to do that comparison. Whereas the embryonic stem cells can differentiate into all cell types. Okay, so your answer has to be like a comparative answer like that, using words like therefore or whereas in the middle. And you're essentially just giving your definition for tissue, then whereas, and then your definition for embryonic stem cells. Okay, and that's how you answer that type of question. Okay, stem cells have been used in both therapy and research. It's been proposed that tissue stem cells could be used to repair severely damaged muscle tissue. Suggest how this might be done. So if we've damaged muscle tissue, then ideally what we're going to be doing is we're going to be taking the tissue stem cells, and we can either grow them in the lab and differentiate them in the lab into muscle cells, and then graft or insert those muscle cells into the muscle tissue itself, or we can directly put the stem cells into the muscle tissue and let them differentiate into muscle cells once they're in the tissue. Okay, so something along those lines would get you the mark for that one. The key thing is mentioning that is going to be muscle cells that will differentiate into, because it'll be muscle cells which will make up the new muscle tissue. Okay, part 2. State how stem cells can be used as model cells in medical research. So you get a couple of choices here. You could say for drug testing or to study how diseases develop. Okay, straightforward knowledge on that. And then part B here, describe how cancer can develop and spread through the body. So again, this is one of those questions where when you see it, you should, if you're feeling a note of dread, that's not a good sign, because that suggests that you have not managed to memorize the content well enough to answer a 3-mark question like this. When you see a 3 or 4-mark question, or even a longer question, if you've done the work and you feel prepared, you should get excited about that, because it's an opportunity for you to show off all of your rote learning knowledge, everything you've memorized and worked hard on your notes on. So for this question, describe how cancer can develop. You're writing basically those bullet points on the cancer section of this topic. So the fact that cancer cells divide excessively, the fact that they fail to respond to regulatory signals, the fact that a mass of abnormal cells will form a tumour. Then on to state that sometimes those cells fail to attach to each other, and they can break away and spread through the body and form secondary tumours. Okay, so if you said an answer like that, you're very much going to get the 3 marks there. Okay, there are 4 different points you can make in the mark scheme there to get those 3 marks. Okay, so cells divide excessively, they fail to respond to regulatory signals, they form an abnormal mass called a tumour, they can fail to attach to each other and then spread through the body, break away and spread through the body, and they can form secondary tumours. Okay, so that is an ideal answer for that kind of question. Okay, folks, so I hope it's been useful to work through these questions and that they have helped you apply what we have learned in topic 1. Okay, good luck with your studying. And if you've got any questions or suggestions, then pop them into the comments and I will get back to you. Thanks, folks, and class dismissed.